The second lecture in the series for Animal Cell Culture Techniques and Application. I am Professor Anita Roy from School of Biological Sciences, Indian Institute of Technology, Delhi, and I will be taking your second lecture in the series. So, if we recapitulate what we already learned last time, so we understood how primary cell cultures are done by taking out cells from an animal, say from a human being or from a mouse, and then that organ is then macerated to obtain single cells. The single cells are then put into a dish for culture. So, that is your primary cell culture. Once the dish is overgrown with cells, then you take out the cells, put them in another dish with the requisite media, growth conditions, and then this forms your subculture or the secondary culture. And if you can keep doing this repeatedly, then this will give rise to a cell line. And the various kinds of cell lines that you can have are continuous cell line. So, if your sample is a cancerous cell, or sometimes if you have primary cells that have been infected with certain viruses, then those cells can keep replicating without undergoing a cessation of division. So, this can give rise to your continuous cell lines. However, normal human cells, diploid human cells, have a finite replicative capacity, and therefore, they would only be kept in culture for a finite period of time, and that gives rise to your finite cell line. Okay, so today what we are going to learn is what are the conditions that are required for cell culture and how do we perform cell culture? What are the things that we require in terms of instruments, in terms of glass and plastic wares, and so on. So, first let us understand what is the physicochemical environmental conditions required to keep your cells healthy and alive. So, the first and foremost thing is temperature. So, if you take out cells from the human body, because the human body has a normal body temperature of approximately 37 degrees centigrade. So, therefore, the human cells would also require similar temperature for their growth and optimum cellular function. So, a temperature of 37 degrees is maintained. So, as you can see here, the first condition, temperature, is roughly approximately 37 degrees, and in cell culture, in laboratories, or even in biopharmaceutical industry where human cells, animal cells are grown, this temperature is strictly maintained. The second condition is pressure. So, we live in on atmospheric pressures. This is an atmospheric pressure condition, right? So, there is no strict pressure condition to be maintained. It is simply atmospheric pressure. So, no strict pressure conditions to be maintained. The third requirement is humidity, and this is of utmost importance. Why? Because when you are growing cells, cells are generally grown in liquid or semi-solid medium, and therefore, when these cells are grown, you need to maintain various other conditions. For example, conditions of osmolarity. So, if you are growing the cells in a liquid medium and the medium evaporates out, then you would be concentrating your solution. You do not want that. So, you need to maintain a humidified atmosphere for the cells to grow. So, in the chambers where the cells are grown, along with the temperature of 37 degrees centigrade, the chamber is also humidified, so that the cells are maintained at a requisite osmolarity too. So, osmolarity. Now, you must know, you must have heard of the term osmolarity, osmotic pressure, and definitely the cells that we grow require to be grown in isotonic environment. That is, because the cells have a selectively permeable membrane that covers them. So, if you grow the cells in a solution which has which is hypotonic, okay, then in a hypotonic solution there would be the cells would be affected. Similarly, if you are growing cells in a hypertonic solution, your cells would be affected. So, you do not want those conditions, you want to grow them in a osmotic pressure condition where the cells are happy, it is an isotonic condition. So, that is maintained through the media and the media constituents. So, what are the main media constituents that maintain this? They include, among other things, the salts present in the media, the glucose that is present in the media, the amino acids that are present in the media. So, they all contribute towards this osmotic pressure of the media. So, typically the osmotic pressure range, as you can see here, is between 290 to 320 milliosmoles per kilogram. And the next important condition to be maintained for the cells is the pH. Now, our body functions primarily at a very narrow pH range. So, that is between pH 7.2 to 7.4. So, this is a very narrow pH range where most of our physiological functions are undertaken at an optimum condition. So, this again has to be maintained for the cells to have an optimum growth and physiological properties. So, this is what we know for the moment, that the ideal pH is 7.2 to 7.4. So, why is an ideal pH required? What is it that would go wrong if you do not have an ideal pH condition? So, all the physiological functions, the functions of enzyme, proteins, all of them, even ion channels, they all depend on this pH. So, for example, if you do not have the right pH, then it could lead to the proteins not having their correct conformation, and that would lead to the proteins not being able to function optimally. Therefore, having a right pH is of paramount importance for the functioning of the cells. So, how is the pH then maintained in the culture conditions? So, they are maintained through maintenance of the pH of the media. So, the liquid medium in which the cells are grown. So, let us have a look at this media. This is a typical example of the media that is used to grow HeLa cells. So, this media is called DMEM, and as you can see, this media is colored almost red. The reason is that there is a pH indicator that is dissolved in the media. So, if the media turns acidic, then the color would turn more yellowish. If it is alkaline, it would turn pinkish. So, this is the media in which your cells are grown, and this media contains, among other constituents, sodium bicarbonate, and the bicarbonate carbon dioxide buffering system is the main buffering system that works to maintain pH of this media and therefore pH of the cells that grows in the media. Now, let us come back to this slide. So, as you can see, the sodium bicarbonate carbon dioxide buffering system is the main buffering system that maintains the pH of the media in which the cells are grown. So, how does this work? So, sodium bicarbonate, being a salt, would ionize in water into its constituent ions, and so therefore, you would have the bicarbonate ions. On the other hand, because you would also have carbon dioxide in the chamber in which your cells are being grown, therefore, the carbon dioxide would also dissolve in water to give you the acid, the carbonic acid, and this would therefore again ionize to give you the bicarbonate ions. Now, if there is a change in pH due to addition of these ions, the protons, into the solution, then the bicarbonate ions would mop up these protons, and therefore, your pH of the buffer would be maintained. So, this is how the sodium bicarbonate carbon dioxide buffering system works to maintain pH of your media in which the cells are being grown. And therefore, because you have already known that bicarbonate, sodium bicarbonate carbon dioxide buffering system works to maintain pH, therefore, you would very well understand that carbon dioxide is required for the growth of human cells or animal cells in culture. So, the carbon dioxide would typically the levels of carbon dioxide provided in the chamber in which your cells are growing would be between 5 to 10%. It depends on the kind of media you are using, and depending on that you can change this carbon dioxide percentage, and this carbon dioxide is required to maintain the pH of the media. So, what is it that the media contains? So, as we have seen before, this is the media in which your cells would be growing. So, other than sodium bicarbonate, what are the other things that your media would have? So, the media would have primarily a carbohydrate source. So, in this case the carbohydrate source is glucose. Apart from that, the media would have amino acids. So, amino acids are the basic building blocks for proteins, right. So, for this reason you would have the various amino acids, essential amino acids, as well as some non-essential amino acids. So, they are listed here, you can see them, it is like histidine, isoleucine, leucine and so on. You would also require fatty acids, okay. So, linoleic acids, etc. Vitamins and cofactors. So, vitamins and cofactors are absolutely essential for the growth of these cells. They are required for various metabolic functionings of the cells such as inositol, thiamine, riboflavin, etc. You need inorganic salts. The inorganic salts like sodium, potassium, calcium, they are not just required for various physiological functioning, signaling processes to take place as well as signal transductions to take place. They also are required to maintain the right pH and the right physiological conditions inside cell. So, inside cell you would have specialized chambers like lysosomes where the pH would be much lower. You would have chambers like endoplasmic reticulum where calcium is stored. Calcium is also stored in mitochondria. Part of the calcium is also stored in mitochondria. So, that is also required for the functioning of mitochondria, functioning of the endoplasmic reticulum. Okay. You would also need some trace elements, for example, selenium. pH indicator. Now, I have already mentioned that a pH indicator is usually added to the media. This is to ensure that you get a ready feedback as to the growth of the cells as well as the change in pH of the media as you are culturing the cells in the media. So, with passage of time you can see if the media is turning yellowish, then that means that your media is being spent and it is becoming more acidic. So, you need to put in fresh media or subculture the cells and put them in a new in fresh plate with fresh media. Okay, so to come back to what are the other things that you would require. So, we have already seen that you would require to maintain the temperature, you would require to maintain a certain percentage of carbon dioxide, 5 to 10%, you require humidity, you require humidified chamber to maintain the osmolarity, you would require media components such as bicarbonate, sodium bicarbonate to maintain the bicarbonate carbon dioxide buffering system. You would require a source of carbohydrate like glucose, you require essential amino acids, lipids, you would require vitamins and cofactors, inorganic salts. Other than that, the cells in our body would also require some growth factors or sometimes some hormones to grow, and these cannot be provided by the media itself. The media is an artificially prepared media. So, instead of that you would either have to provide these components one by one. Say if you know that the media, the cells would require insulin to grow, then you provide a little bit of insulin, adequate amount of insulin to the cells to grow. If you know that your cells require albumin to grow, you would have to provide albumin to the cells to grow or transferrin for the cells to grow. So, but to bypass all of these, in general we use something called serum. So, serum is either taken from bovine or from calf, and then this serum is mixed with the media. And what the serum does is it provides these essential nutrients, essential growth factors, along with providing of course amino acids, lipids, etc. So, the main function of the serum is to provide these growth factors, the albumin, the transferrin, some amount of iron to the cells for the cells to grow and function correctly. It will also of course provide amino acids, salts, lipids. Now, finally, because this media in which the cells are grown is such a rich media, there is always a chance of contaminating the media with bacteria. Now, as we know that there are a number of bacteria right now, so the air that I am breathing would also have bacteria, would also have fungal spores. So, this bacteria can contaminate the media in which you are trying to grow your cells, and in order to prevent the growth of the bacteria at the expense of growth of your cells, we add antibiotics. So, typically antibiotics such as penicillin and streptomycin are used in the media to prevent contamination of the media with bacteria. Okay, so let us recapitulate. So, to keep the animal cells healthy, alive and functioning to their optimum physiological function, we need to maintain certain conditions. Those include number 1, temperature at 37 degrees maintained in a humidified incubator. That is very important, a humidified incubator. Incubator, by the way, is the chamber in which we will be keeping these cells. So, that chamber, the essential function of that chamber, which I would be describing later on in this lecture, is to maintain the temperature, the humidity as well as the carbon dioxide percentage in the percentage, so that the cells have an optimum condition to grow in. So, along with the temperature, we need to provide media with amino acids, glucose, lipids, vitamins, inorganic salts, trace elements. We also need to, because of the media, we can provide it with a pH of around 7.4, and this is maintained with the help of this bicarbonate carbon dioxide buffer system. Along with this, we also have to add some additives such as serum, okay, that would provide essential growth factors, hormones, lipids, amino acids. Also, we add antibiotics to prevent growth of bacteria. All of these together will give you happy cells. Now, here's some facts. So, we have learned that human cells require a temperature of 37 degrees centigrade, a carbon dioxide percentage of 5 to 10% for growth, and they have to be kept in a humidified moist incubator so that they are functioning correctly. But it is interesting to note that insect cells, which are often used in biopharmaceutical industries, do not require this kind of a carbon dioxide percentage requirement. So, they can grow in an incubator where the temperature is maintained at a much lower temperature, around say 25-27 degrees, and they do not require this carbon dioxide exchange. So, they are much simpler cells to grow, but they are very, very versatile, because unlike bacteria, which do not offer us much prospect in terms of complex glycosylation, insect cells can somewhat glycosylate. They have the glycosylation mechanism because they are eukaryotic cells. So, they have glycosylation mechanism, and therefore, many proteins are produced in insect cells. They can be also correctly glycosylated in insect cells. So, here is an example. So, the human papilloma virus, it is a virus that causes many cancers in humans, including adenocarcinoma. So, this human papilloma virus, the vaccine against the virus, is actually developed in these insect cells. So, now you might ask, then, is it that the human virus actually grows in insect cells? Well, no, the human virus does not grow in insect cells. What the biopharmaceutical industry does is that they just take the protein against which the protein of the human papilloma virus, against which our bodies can produce antibody, right? And therefore, if they can produce antibody, then we are safe. So, if you take that protein of the human papilloma virus and if you put it in the insect cell, then the insect or the DNA for this protein can be put in the insect cell. The insect cell will produce a lot of these proteins, and therefore these proteins can then be used to immunize humans. Alright, so far we have talked about the conditions in which animal cells are grown, the kind of media they grow in, what are the constituents of the media, the temperature, pH requirements, how they are maintained. Now we would go into what kind of media, the state of the media that we use in. So there are two kinds of media mostly that are used in that sense. So one is the liquid media. So the example here that we see here is actually a liquid media. If you see this, it's a liquid media. But you can also have a semi-solid media. And a semi-solid media is essentially the same kind of media in which you would add collagen. So collagen would form a gel-like substance, and therefore the media would turn into a semi-solid gel. And what this does is that it helps to immobilize cells. And therefore, if the cell is immobilized and it starts dividing, then the number of divisions of the initial cell would correspond to the size, the final size of the cluster that it forms, right. If you have one cell that has divided 10 times and another cell that is able to divide 1,000 times, right, then the size of the cluster would be different. One would be smaller, the other would be bigger. So this is exactly what is used to understand if a certain cell has a cancerous propensity or not. Because if a person has cells that have a cancerous propensity or that are cancerous, then those cells would divide indefinitely, and also they would have a higher number of divisions, they would be growth factor independent and so on and so forth, and therefore those clusters that they form would be larger in size. So semi-solid media is very much used to enumerate this kind of abnormal cells. Okay, so now we are going to look at the kind of equipments that are used in cell culture. So the first thing that we are going to look at is the biosafety cabinet, also called the cell culture hood. So why do we primarily need a biosafety cabinet or cell culture hood? There are two functions of this biosafety cabinet or cell culture hood. One is to protect the cells that we are growing from external environment. So what does the external environment have? As I explained to you, the external environment would have bacteria, would have fungal spores, and we do not want to grow bacteria or fungal spores. We only want to grow the cells. So we have to protect the cells from those bacteria and fungal spores. At the same time, if we are working with cells which are pathogenic, or if we are working with some viruses, and we do not want to contaminate ourselves with them, we do not want to be infected with them, we also have to protect ourselves from those cells. And therefore, this biosafety cabinet or the hood acts as a barrier in both the cases. And how does it work? So the primary means by which it works is that it works on the principle of a laminar airflow. So what is a laminar airflow? So laminar airflow would be one where the flow lines are of the airflow is absolutely parallel. Okay, there is no turbulence, it's absolutely parallel flow lines. And the second thing that it uses are the HEPA filters. Now you must have heard of HEPA filters. Nowadays in India, many cities are reeling with pollution, air pollution, and therefore many of us may have filters at home to filter out air, and those filters actually use HEPA. So these high efficiency particulate air filters are capable of removing particles such as bacteria, and therefore these filters are installed in the laminar hood or the laminar biosafety cabinet. So if this is the biosafety cabinet, you would find a set of these HEPA filters installed at the top here, and the air would flow from the top to the bottom and would be circulated back. These chambers or hoods or cabinets are also equipped with UV lamp, and the UV lamp is used to initially sterilize this chamber. So typically when people work on these laminar hoods or filters, so at the start of the work they usually turn on the lamp and leave the lamp on for 15 to 30 minutes, and then they start their work. This is to ensure that the chamber inside is absolutely sterile. Now, depending on the class of biological material being used inside the hood, you would need different categories of biosafety cabinet. So, they are generally categorized as class 1, 2 or 3, and depending on the severity or the infectivity of the reagents of the biological agent being used, you need a higher class of biosafety cabinet. The second thing that we are going to discuss today is an incubator. So, we have already learned that an incubator is required to maintain the correct temperature, the correct percentage of carbon dioxide as well as a humidified chamber, right. So, these are all attained in this controlled environment inside what is known as the incubator. And the incubator maintains this temperature of 37 degrees. It is capable of maintaining a carbon dioxide of 5 to 10%, and usually water is left in a tray at the bottom of the incubator, and that helps in creating this humidified chamber. So, very often these incubators are also programmed to undergo a sterilization or a decontamination process. So, that would remove any bacteria or fungi that might be present inside. So, cells are grown inside these incubator, and as I explained to you, the cells in the incubator are maintained in a sterile condition and at 37 degrees centigrade, and also the carbon dioxide is maintained inside the incubator. Sometimes to grow certain kind of cells you might also need to regulate oxygen conditions. So, if you would like to grow cells at a hypoxic conditions, that could also be maintained in the incubator. Alright, so you are growing cells, you have cultured cells in the hood, in the laminar hood, you have kept them to grow inside the incubator, but how do you visualize the cells? You need to visualize cells to understand if they are happy, if they are healthy, how far they have grown, how many they are, do you need to subculture them, do you need to change the media, and so on and so forth. So, to do that you have to visualize the cells, and that is done under a microscope, an optical microscope. So, on the left you can see the typical image of a microscope, and you would see that this is a little different from perhaps the microscopes that you have used in your schools, and the reason is that this is an inverted microscope. So, why is it an inverted microscope? Because you can see that the light path, the sample stage and the lenses, right, the objective lenses, they all are in this fashion. Your school microscope would have had the objective lenses right here. Okay. So, why is this kind of a configuration important? It is important because you would have to observe your cells that are growing in certain chambers under the microscope. You do not want to take out the cells and put them on a slide and then visualize under the microscope. You want to visualize them as they are growing in the chambers, and therefore, you would need this kind of an inverted microscope configuration where you have the possibility to put your samples very close to the objective lens, which is right here at the bottom, and you can put your samples here, the flask or the plate on which you are growing the cells, right on the stage, and then you are able to visualize them. So, typically the magnifications that are used, the objective lens magnifications that are used are of the order of 20x or 40x, and that is enough for you to visualize the cells that are growing in culture, as well as you can also detect if your cells or the media in which you are growing the cells is contaminated. So, if there are bacterial particles or if there are fungal contamination, you can also observe that under the microscope. Okay, then the other piece of instrument that you would require is a centrifuge. Now, what is a centrifuge and why you require it? So, on the left you can see the image of a centrifuge, and on the right you can see a cartoonic representation of what it actually does. So, you have your sample. So, the sample are actually cells that are floating in the media, and you would want to get all the media out from these cells and just get the cells. So, if you want to do that, the first thing that you have to do is centrifuge the cells. What this does is it increases the force on these or makes these cells heavy. So, once these cells are made heavy, they actually would precipitate out. So, to increase that kind of to make the cells heavy, you actually need to increase the G, and that is done using this centrifuge. So, by high speed rotations, you would be able to pellet down the cells. So, the word that we use is pellet. So, this is a pellet where your cells have actually sedimented out. So, it is a sedimentation process, but at high speeds of rotation, and this would enable you to remove the entire media while retaining the cells. And this process is often used to replenish this the media. So, you do not want to carry over the spent media, or you might want to add in fresh media that has a different component, and you would like to understand the effects of the component. So, for all those purposes you would require the usage of such kind of a centrifuge. Okay. So, now let us look at the vessels in which you would be typically growing your cells. So, on the right you see the image of certain vessels in which your cells are growing, and I am also going to show you some of these apparatus. So, here is what we call a flask. So, this is one of the typical cell culture equipment or plasticware in which you grow the cells. So, we can add the media with the cells into this flask, and the flask actually has a growth surface which is this. So, this part of the flask, is actually the growth surface. So, you add about 10 milliliters of your media with the cells, and you keep the flask in the incubator, and that is how you can grow the cells. So, you could also grow the cells in small plates like this. Okay. Again, you have the plates are of this kind, and you have a growth surface for the plate, and you grow the cells in the plate with media inside the incubator. So, when you are culturing cells, when you are using these, we are using these in the biosafety cabinet or the laminar airflow. So, when we culture the cells, when we add cells to these flasks, or we add the media, we add the constituents, everything is done inside the biosafety cabinet or the laminar hood. The reason is that we would like to protect the cells from contamination. We would also like to protect our cells from any contagious agents that we might be using, such as viruses. So, but once these are cultured, we have to put them into the incubator. So, these flasks or the plates are then put inside the incubator once you have cultured them. Alright. So, the flasks or the plates that we generally use, so initially they were all made of glass. They were made of glass and they were coated with certain proteins such as fibronectin or collagen that would ensure that the cells the human cells are able to attach, adhere, and they have a growth surface. As I explained, this is very important for the cells to grow under normal physiological conditions. You know, it is a replication of the normal physiological condition. But slowly we have shifted to plastic or polystyrene, polypropylene plates. So, these actually these plates are in general very hydrophobic. So, they have to be turned hydrophilic. Also, they are sometimes additionally coated with proteins such as fibronectin or collagen to provide that kind of an adhesion surface for the cells to grow. Alright, so we have grown the cells, we have used flasks or plates to grow the cells in the requisite media with supplements such as serum, we have added antibiotics, we have provided the right kind of buffering system in the incubator with 5 to 10% carbon dioxide, temperature of 37 degrees, and the cells are growing happy. But we would also like to store the cells and use them in future. So, how do we store the cells? So, the main method by which we store the cells is called cryopreservation, and this is a technique by which we freeze the cells at ultra low temperatures, and this would be done in such a manner that it maintains the integrity of the cells. We do not want to affect the integrity of the cell membrane or the organelles. We want the cells to revive properly once they are thawed. So, for this certain precautions have to be undertaken when we are doing the cryopreservation of cells. So, as I mentioned, the cryopreservation of cells or tissues is undertaken at ultra low temperature, and the main consideration is to keep the cells intact so that they can be revived and they will survive once they are thawed. And the storage temperature in general that is used is somewhere around minus 180 degrees centigrade, and this is achieved through liquid nitrogen. So, the storage is done in liquid nitrogen. But the cells are not immediately put in liquid nitrogen. The storage has to be done in such a fashion that the cells slowly attain a temperature of minus 80, and then they are transferred from minus 80 to minus 180 degrees centigrade. And this transition from room temperature or from 37 degrees to minus 80 has to be a very gradual freezing process. So, the change in temperature or the decrease in temperature has to be of the order of 1 degree per minute, and this is to ensure that very large ice crystals do not form, because ice crystals will damage the cells, damage the integrity of the cells. So, to do this, in order to achieve this, cryopreservatives are used. Cryopreservative, the main cryopreservative and the most widely used cryopreservative is called dimethyl sulfoxide or DMSO. And typically we use about 10% DMSO and we resuspend the cells. So, as I explained, centrifugation is a process by which you can remove the media while retaining the cells. So, if you centrifuge the cells that were growing in the media, you will be able to obtain the cell pellet and you can remove off the excess media. Now, if you resuspend these cells in serum with 10% DMSO, this will help you to keep the cells happy while they are undergoing the freezing process. So, you need some amount of albumin or proteins in the freezing mixture that is provided by the serum. You also require cryopreservatives such as DMSO. So, other than DMSO, there are some other cryopreservatives used. So, glycerol is another cryopreservative which is used very often for bacterial preservation. You can also use sorbitol, etc. But as I mentioned, dimethyl sulfoxide is the most commonly used cryopreservative agent. Okay, so now you have frozen your cells. So, but now you would want to take the cells out of liquid nitrogen and grow them. So, how do you do that? So, you take so for freezing we were doing a gradual freezing process, right? But when you defrost the cells, when you want to revive the cells, this process has to be a quick process. So, you take out the cells from liquid nitrogen, the vials in which you have frozen the cells, you take them out from the liquid nitrogen and you thaw them quickly at 37 degrees in a water bath. So, once that is done, the next step is to remove DMSO from the cells, because DMSO also has other effects on the cells which you do not want. They have some detrimental effects on the cells. They can also start differentiating, or they can change the type of the cells that you're growing. They might acquire a little different morphological feature which you do not want to do. So therefore, again you have to centrifuge the cells. When you're doing that, when you have your cells in serum plus DMSO, we dilute that in media, centrifuge the cells, you have pelleted the cells down, you take that pellet, resuspend in media and put it in a flask or a plate and grow the cells. So it's a simple process, but the trick in freezing and in defrosting is that the freezing has to be a slow gradual process. It requires a cryopreservative such as DMSO, and the frozen cells have to be stored at around minus 170, 180 degrees centigrade in liquid nitrogen for long time storage. And long time means you can store the cells 10 years and beyond. Whereas when you are defreezing the cells, that is a quick process. You have to put the vial at 37 degrees, you need to thaw the cells and centrifuge the cells to completely remove the cryopreservative. You do not want cryopreservatives to be present when you are growing the cells, and then you put the cells back into the flask or the plate, and then put the plate or the flask with the media, the cells into an incubator where you are maintaining the right temperature of 37 degrees, you are maintaining the carbon dioxide percentage, you are also maintaining a humidified atmosphere. Alright, so I have showed you flasks of this size. You can, if you would like to grow more cells, you can increase the size of the flask. You can also increase the size of these plates. But there is a limit to which you can increase the size of the flasks and the plates. And why is that limited? That is limited because the more you increase the size, the less would be the diffusion capacity of, say, carbon dioxide or some essential nutrients, and therefore you have, the more you increase the size of these plates or the flasks, the main difficulty is in terms of diffusion of carbon dioxide or replenishment of media, because you do not want to produce pockets of cells that have seen a different physicochemical environment than the rest of the cells growing in that plate. So then how do we scale up the process for biopharmaceutical companies? Because biopharmaceutical companies would require to produce, say, large amount of hormones, large numbers of antibodies, and so they would require large cultures. For them, a 10 ml, 20 ml or even a 100 milliliter culture would not be enough. So for that you need special flasks, special bottles to culture the cells, as well as special incubators. Okay. So why we need it? Because for industrial application process there are critical components to be controlled and that are absolutely must to be controlled, and we have already learned why. So media pH, dissolved carbon dioxide and oxygen, these are parameters that have to be strictly regulated and controlled. Otherwise, from one batch to the next, you would have variations that you normally do not want to have. Say batch 1 has produced 1 milligrams per milliliter of a component, the other batch would produce, instead of 1 milligram, it is producing 0.01 milligrams, and you do not want that. So you do not want these batch to batch variations, you want it to be very uniform. So these parameters have to be tightly regulated. So therefore, we simply cannot take a bigger and a much bigger and a much bigger flask. There are limitations to that, and the limitations all arise due to diffusional limitation. So here are examples that are generally used in the biopharmaceutical industry where they require to grow large numbers of these cells. So on the left you can see what is called a roller bottle. So these bottles actually have cells coated throughout the surface. So throughout this lateral surface you would have cells adhering to them, and you have media inside the bottle. Now if you slowly rotate the bottle, then the cells at any given time would have a thin layer of media coating them. And because you are rotating the bottle, you are maintaining diffusional properties, you are maintaining the same pH, the same carbon dioxide, oxygen conditions inside the bottle, throughout the bottle. So, there are not some cells here which see a different condition than the cells at the bottom, because you are slowly rotating the bottle. So, these bottles are called roller bottles, okay, and they have cells growing throughout their lateral surface. So, it ensures that there is exchange of gases, it prevents formation of media gradients or components of the media, gradients of them inside the culture. Therefore, provides a very uniform growth condition for the cells. So, this is for adherent cells, so cells that would normally adhere to the surface. But if you have cells that do not require adherent conditions, suppose the cells are the cells of the blood. So, in general, cells of the blood, they do not require an adherent surface. In that case, you can grow them in something called a spinner flask. So, here on the left you can see what we know as the spinner flask. So, what do we have in a spinner flask? The main component here, as the name suggests, is this central shaft that is connected to a magnetic, not a blade, but a magnetic agitator, a paddle. And so, if you put this flask on top of another magnetic plate which has a magnet that rotates, then this being magnetic would also rotate, and if this rotates, then this will agitate the media, and therefore, with this agitation, the media components, there would not be a gradient of the media components, there would not be a gradient of pH inside, because you are mixing up the cells, mixing up the media, and this is a good way to grow suspension cultures. If you have a look at the flask, you can see that there are two inlets or outlets in this flask. So, one of this inlet, one of this which we will call an inlet, would be used to add fresh media into the flask. The other outlet would be used to remove off spent media from the flask, spent media and cells from the flask. So, this is a very nice way to not just ensure that the cells are growing in uniform condition because you are agitating the media, but you also have different inlets and outlets that would ensure that you can add fresh media into the cells while removing spent media and cells from the flask. But what if you would like to use the same apparatus for adherent cells? So, for adherent cells, you may be able to use the same kind of apparatus provided you have the cells growing on something called a microcarrier beads. So, suppose you have beads that are suspended in the media and the beads are coated with your cells, then definitely you can repurpose this spinner flask to grow your adherent cells. So, here is an example of these microcarrier beads. On the left you can see an image of the beads. So, these circular spherical ones are the beads and the ones that are marked with the red arrows are the cells. So, the cells can grow adhered to these beads, and the beads are generally made of some polymeric material, and they can be kept in suspension by the rotation of the paddle, the central shaft. Okay. And therefore, you can use these beads in combination with your spinner flask to grow adherent cells in culture in very large numbers. Okay. So far we have looked at what are the requirements for the cells to grow, what are the conditions, physico-chemical conditions, what are the equipments that you need, what kind of glasswares or plasticwares, what kind of flasks that you need to grow the cells or plates that you need to grow the cells. What if you need to grow the cells in much larger numbers, then you can go and use a roller flask, or you could even use a magnetically rotating central shaft in a flask, and these can provide you with much larger numbers of cells. And for these, again, you would need special incubators, because the normal general incubators may not be sufficient to accommodate the kind of extra equipments that you need. Say for the roller flasks, you need something to rotate the flasks. For the spinner flasks, you would need a magnetic stage on which the spinners can rotate. So these will be extra components that should be provided inside the incubator. Now let us have a quick look at the growth curve of the cells, because we have so far looked at how to grow the cells, but how exactly do the cells grow and what is the mathematical formulation for this. So if you look at the graph that is shown here, you would see that the cells initially, the growth of the cells initially is slow. Okay. So as I explained the last time, when you put the cells into culture, they take time to adhere, they take time to sense their surrounding environment, and they then grow slowly. But once they are comfortable in their new environment, then they actually grow at their general growth rate. So that growth rate can be seen at this log or exponential phase. So initially it is called the lag phase, and then the cells grow exponentially in the log phase. So now the cells may have grown to an extent that they have spent the media. So there is no more, there is not enough glucose to support their growth, or maybe they do not have enough surface to grow on. Then they would go into a stationary phase where their numbers may not change much. And if you still keep growing them in those same flasks, then you would see that their numbers would decline and they would go into something called a death phase. So this is the general growth curve, what we call the growth curve of the cells. If you have another look at this curve, you would see that it is versus time. So on the y axis we have plotted the number of cells, and on the x axis it is versus time. Okay, so you are looking at the number of cells with time. But if you think of the way the cells were growing in the exponential phase, and if you understand, and if you now try to connect it with how the cells grow. So if you have one cell, it would divide to produce 2 cells, right? 2 cells would divide to produce 4 cells, and so on and so forth, right? So if you understand the number of generations that have passed, say the number of generation is G, okay. So if it has passed 1 generation, then it would be 2 to the power 1, which would be 2 cells. If it is 2 generation, then 2 to the power 2, 2 to the power 3, and so on and so forth. But instead of having 1 cell initially, if you had n0 number of cells, that is the initial number of cells, then the total number of cells after certain number of generations would be given by this formulation, where n is the total number of cells after G generation, n0 is the initial number of cells, and therefore this formulation would give you roughly the number of cells after G generation, taking into consideration that you have very little cell death, right. Now there is another, so here we were talking in terms of generation, right. If we think that in terms of the time, so it is doubling time. So the time required for one cell to produce 2 cells, and this is a very characteristic time for different cell types. So some cells have a doubling time of 18 hours, some cells may have 24 hours, some cells may even have about 2 days. So this doubling time is very characteristic of the cell, and if you know the doubling time, you can still estimate the final number of cells that you would get. So instead of saying G, if you replace G with the total time that it has taken or that has passed from the initial point of seeding your cells, and you divide that by the doubling time, then that itself is G, would give you the number of generations, and therefore you can convert this equation into something like this. So all of this would help you to estimate the total number of cells that you get at the end of your culture. Alright, so finally let us look at characterization of cell lines. So we have talked about how cell lines are established, right, but you would before you can start using those cells, you need to characterize them, you need to understand how they look. For example, here you can see there are two kinds of cells. On the left you have fibroblast, on the right you have the classical HeLa. You can very well appreciate the difference in morphology between the two cells. They are both adherent cells, but they don't look quite the same. So, the first thing that you have to understand when you are characterizing the cells is to understand what is the origin of the cell, what is the history of the cell, where did it come from? Did it come from mouse, did it come from human, did it come from a cancerous tissue, did it come from a normal tissue? Okay. How does it look? How does it look morphologically? Is it adherent? Is it suspended? Okay. What are the growth kinetics? How fast does it grow? What is the doubling time? So, these are the things that you would record when you are characterizing the cells. You would also want to know the karyotype of the cell. What is karyotype? So, we know that we all have 23 pairs of chromosomes, right? 22 plus 1 of the sex chromosomes into 2. So, on the left here you can see the typical image of a karyotype. These are the chromosomes in our body, typical chromosomes in our body, all the 22 pairs of chromosome plus the sex chromosome. And on the right you can see the karyotype of a HeLa cell. Now, HeLa cell, as I had explained, that this is a cell line that is obtained from a cancer, and cancerous cell lines often have abnormal karyotypes. So, you can see the abnormality in the karyotype. So, if you ever have a cell and people claim it is a HeLa cell, you can go back, look at the morphology, look at the doubling time, look at the karyotype to verify whether this is indeed HeLa or not. So, other than looking at the karyotype, there are other things that can be done. You can see whether the cell line is pure, whether it is contaminated with virus or bacteria. You can also see its response to growth factors. So, normal cells would require certain growth factors to grow, They might require insulin, they might require some other growth factors, whereas cancerous cells often do not require external growth factors to grow. So, all of these together would help you to characterize cell lines, and characterizing cell lines is very important before you start to use it. You have to know the type of cells that you are using. Alright students, that is all for today. Next time we would look further into how cell culture is used in for modern biological applications. Thank you.